Imagine a giant spaceship lands in your backyard. The door flings open and you are invited inside to see what you can learn. Now, the technology clearly is millions of years beyond what we can make right now. Well, this spaceship is biology here on Earth. Life itself, in fact, is a technology that has been evolving for billions of years. And in this video, we will explore how it works and discover some of the amazing things that it could enable in the near future. So since the start of the global coronavirus pandemic, I started a completely new adventure into the fields of computational biology and protein engineering to try and understand how these complex nanomachines function. And after working in this field for quite some time now, I wanted to share some of the amazing things I've learned because my audience of computer science people, which is you guys, are in a unique position to make incredibly valuable contributions to one of the most promising areas of modern science. So this series will contain several parts. In this first video, I want to start by building up a general understanding of the physical processes that underpin our biology, going from the genetic code embedded in our DNA to the insane world of proteins, covering the basic mechanics of the coronavirus, and introducing emerging fields like AI-assisted computational biology. Now, I'm doing this because you can't really dive into the machine learning aspects of this field without at least having a basic understanding of the biology that's involved. I mean, I learned a ton of fascinating stuff over the past year, and so I try to condense as much of that as possible into this video. The next videos then will focus on specific machine learning techniques and architectures that are becoming ever more prominent in this quickly evolving field. We'll look at various challenges like, for example, property prediction for a given molecular structure, the famous protein folding problem in DeepMind's AlphaFold, and introduce the exciting emerging field of AI-driven molecular design. So if you're eager to set your first steps into one of the most promising areas of modern science, I hope you're ready to dive in deep. My name is Xander, and welcome back to Archive Insights. So what are proteins and why should I care? Well, the very first thing that I'd like you to contemplate is that you and I, in fact, every living biological entity on this planet, is in essence an incredibly complicated and interconnected molecular machine. And what I mean by this is that every process inside our bodies, no matter how complicated or how magical it may seem, they're all governed by the same laws of nature that we study in fields like chemistry and elementary physics. And now, the reason that these simple natural laws give rise to the enormous complexity of life is that over billions of years of evolution, the tiny atomic particles that make up our universe, well, they started coalescing into ever more complex formations that gradually diversified and started interacting with one another. Now, there are many open questions in this field of evolutionary chemistry, or abiogenesis, as it's called. But what we do know is that at some point in the evolution of life, simple organic molecules started forming small self-replicating systems, which then became subject to random mutations and natural selection. And over billions of years, these systems gradually diversified and produced more familiar biological elements like cell barriers, genetic material, and the ability to capture ever more energy from their environment through processes like photosynthesis, for example. Now, to get an even richer appreciation of this miracle of life, I think it's useful to get a sense of scale with respect to the concepts that we'll be talking about in these videos. So if we start out with a small object of familiar size, let's say a coffee bean or a grain of rice, that measures several millimeters across, and we gradually zoom in, well, we soon encounter things like amoeba, which are fairly large but unicellular organisms. Going deeper, we encounter things like the human egg cell and sperm cell, measuring several micrometers across. Next, let's dive inside these cells and look at what they're made of. Here we find things like the chromosome, which is essentially a huge storage container for our DNA inside the nucleus of every living cell. Next, we find things like bacteria, for example here the mitochondria, which are like the power plants inside of our cells. They burn sugar molecules to produce ATP, which is the main unit of chemical energy that is used to power all other processes going on in the cell's interior. And if we zoom in even more, we have to start measuring things in nanometers. So this is already about 1,000,000 times smaller than the millimeter size of the coffee bean that we started from. Here we encounter viruses that are so small that they can easily penetrate into our cells and start affecting their function by injecting their own genetic material into the machinery of the cell. Essentially, they're hijacking our own molecular factories to produce copies of the virus. And in this sense, the computer analogy is a pretty good one. The virus doesn't have any hardware to produce copies of itself. Its only way to spread is to infect the host cell and use its hardware to make copies of itself. And finally, if we zoom in beyond the scale of the smallest viruses, we encounter the domain of proteins. Proteins are essentially small nanomachines that perform very specific functions inside of living organisms. Here, for example, you can see a depiction of what's called a ribosome. It's a complex molecular machine found in all living cells that essentially translates the genetic code written in our DNA and uses it to produce new proteins. But before we take a closer look at how exactly all of this works, let's first finish our biological zoom all the way to the bottom. Here's another protein called hemoglobin. And its function is very simply to capture oxygen molecules in our lungs and transport them across the body, releasing them inside our cells where they are needed for breaking down the sugars in the mitochondria that we just saw. And then finally, if we dive below the nanometer scale, we arrive at the very smallest biological compounds, like glucose and adenine, that are made up of just a few individual atoms. So the main thing to remember here is that all of biology is a tremendously complex hierarchy of interacting systems, each of which is following the laws of nature, but gaining complexity as we move up the scale ladder. In theory, with an infinitely powerful computer, you could solve all open questions about biology by simply simulating a living system down to the atom level. So now that we have an idea of the various components that play a role in this molecular dance, let's take a look at some of the most fundamental processes in this play of life. Specifically, we'll start by taking a look at how exactly the genetic code embedded in our DNA contains the executive program on which all of these processes run. So buckle your seatbelt as we dive into the most fundamental process that powers life on Earth. So deep inside the nucleus of every cell in our body, we find the chromosomes. They're essentially huge storage containers for our genetic material. Humans actually have 46 of these chromosomes, which together contain what is called the human genome, or in other words, the molecular blueprint that contains all the information that you need to build a human body. Now, the genetic material inside of those chromosomes is tightly wrapped around proteins called histones to keep things nice and tidy. If it weren't for those histones, the long DNA strand would become a completely tangled mess. Now, what happens when our cell needs to build new machinery is that a specific section of this DNA, called a gene, will be read for instructions on how to produce a specific protein. The whole process starts with an already existing protein called RNA polymerase that latches onto the DNA and unwinds the double helix into a flat section of genetic code. Using its complex machinery, it will then start to copy the instructions encoded in the DNA sequence in a process called transcription. Now, in this phase, each letter of the gene is faithfully copied over into a complementary chain of base pairs that essentially contains the same information as the gene itself. The result of this transcription is a chain of messenger RNA, which contains the relevant part of the genetic code that's needed to create a specific protein. And once this is done, the messenger RNA moves out of the nucleus and travels into the cytoplasm of the cell. There, it encounters another type of proteins called ribosomes, which are like the protein factories of our cells. So where the DNA polymerase are the guys that figure out exactly how to build proteins, the ribosomes actually get the job done. So the ribosome binds to this messenger RNA and it starts reading its instructions in order to produce a new protein. Now, the fundamental building blocks of proteins are what are called amino acids. These are small molecular compounds that come in unique flavors, and they consist of only about 10 to 20 atoms. So these things are really, really small. In fact, there are exactly 20 different types of amino acids in ordinary biology, which you can think of as the essential Lego pieces that biology uses to make stuff. It's like the fundamental alphabet of life. Freely floating around in the cytoplasm of the cell, every one of these amino acids is connected to a piece of transfer RNA, which contains a unique identifier at the bottom that perfectly matches a specific 3-letter sequence in the genetic code. The ribosome reads the messenger RNA 3 bases at a time. And as each triplet is read, the connected transfer RNA delivers the corresponding Lego block to a growing chain of amino acids that exits the ribosome at the top. And after the last amino acid has been added, the purposefully ordered chain is then released into the cytoplasm of the cell's interior, which is essentially full of water molecules. There, it is subject to a complex interaction of physical forces, both among the amino acids in the chain, as well as with the surrounding water molecules. And this finally causes the entire chain to fold in on itself and to form a complex but unique 3-dimensional structure, which is called a protein. And that's why this entire process is called translation. Because through this beautifully complex mechanism, the molecular engine of life is able to translate a piece of genetic code into an operational protein that can now go and take up its own proper function somewhere else in the body. Alright, so we've just seen that the ordering of the DNA letters in the gene that we start from controls the ordering of the amino acid sequence in the protein chain. And after that, the laws of physics cause that chain to fold up into the lowest energy state that it can reach. In fact, this is exactly what people mean when they talk about protein folding. And it's exactly this task that DeepMind's AlphaFold model addresses. Essentially, DeepMind trained a carefully designed neural network architecture to predict the 3-dimensional shape of a protein, knowing only its amino acid sequence. They did this using a very large dataset of known protein structures, which is called the Protein Data Bank, or PDB. And given that this approach recently started to work amazingly well, it's actually opening up an incredibly exciting area of AI-assisted protein design, which is the space that I'm working in right now. But we will talk a lot more about that in future videos. In fact, once DeepMind eventually releases their academic AlphaFold 2 paper, hopefully with code and models included, I intend to make an entire video dedicated to protein folding. Alright, fair enough. So a protein has a unique shape that's entirely defined by its amino acid sequence, which is coded up in our DNA. But why should that have anything to do with a protein's function, you might ask? Well, a good intuitive way to think about this is to imagine proteins as small 3-dimensional puzzle pieces that fit into certain pockets but don't fit in most others. Additionally, many of these proteins are not really fixed, rigid structures, but they can actually change shape depending on what they're chemically bound to. To give a concrete example of this, let's take a look at a very specific pair of 2 proteins that absolutely blew my mind when I first learned about them. So I'm talking about the motor protein pair consisting of dynein and kinesin. And the main function of these motor proteins is to move stuff around inside of the cell. So it turns out that pretty much every cell in your body contains what are called microtubules, which are essentially long cables that are stretched between opposite sides of the cell barrier. Now, These microtubules, they function as rigid structural highways that the dynein and kinesin proteins interact with in order to move stuff around in your cells. And the way this works is that kinesin, for example, consists of two identical motor heads at one end and a long and flexible organelle at the other that attaches to the cargo that's being moved around. Now, the shape of the motor heads is such that when it comes into contact with a microtubule, it establishes a strong chemical bond which releases a small ATP molecule from the now attached head. And almost instantaneously then, this empty binding site is filled up by an ATP molecule which triggers a shape change of the motor head, causing the other leg to be thrown forward and also bind to the microtubule. And when this happens, it triggers a chemical reaction in the first head, which causes it to let go of the microtubule, and the process loops back from the beginning, essentially causing the kinesin motor to start walking along the microtubule track with its two alternating legs and pulling along anything that's attached to it. I mean, isn't that just absolutely incredible? And these proteins actually do a lot more than just carry around stuff within cells. In fact, similar kinds of proteins are responsible for any kind of muscle contraction in your body. I mean, just think about this for a second. Every time that you move a muscle in your body, millions of these tiny molecular engines, purposefully designed by the genetic code in our DNA, instantly react to an electrical signal coming from your brain, and they start changing shape and physically interacting with other proteins, causing your muscle fibers to contract and your body to move. Also interesting is that relative to their size, these motor proteins move about as fast as an actual car on a highway, but they are 4 times more efficient than your car in converting chemical energy into motion. So you see, I mean, we still have a lot left to learn from Mother Nature. So what you should remember is that the driving mechanism of protein function is the chemical interactions that they have with other substrates in the body. And the way that these interactions happen is largely controlled by their shape. So to give a second example of why shape is so important in the protein world, let's take a look at the biggest biological nemesis of recent times, the coronavirus. So the coronavirus is a single encased cell surrounded on the outside with the so-called spike protein, which gives the virus its crown-like look and hence its name. Now, because of its shape, this spike protein has a particular affinity to binding with a specific protein in the human body, which is the ACE2 receptor on the surface of human lung cells. So what happens is that the virus is just roaming around in the body until it encounters some lung cells, where the spike protein then binds with this ACE2 receptor. This then triggers a protein-powered shape change whereby the viral RNA enters the human cell and it hijacks our own cellular machinery to start making copies of the virus. Fortunately, the protein mechanism that makes viruses so dangerous is also their weakness. Think about the capsid on the outside as a 3-dimensional fingerprint that uniquely identifies each type of virus. In fact, this is exactly how our body is able to mount a defense against a virus attack. By purposefully manufacturing antibodies that bind to the capsid of a specific virus, our adaptive immune system can create a highly targeted defense. And if you're lucky, your immune system will remember how to make those specific antibodies for many years to come, which is precisely what we call being immune. This is also how a lot of vaccines work. You simply manufacture some of those capsid proteins in a lab, and you inject them into the patient without an actual virus being present. The immune system will detect this strange compound and it will start creating specific antibodies that bind to that protein signature so that when the virus actually comes around, our body can mount a highly specialized attack almost instantaneously. So I think by now it's becoming clear why AI models like DeepMind's AlphaFold have such an incredible impact. on areas like drug development and vaccine design. Because if we can use AI to predict the structures of most important proteins in biology and model their interactions, well, this gives us amazing new insights into how they function. Looking at the future then, many people agree that this is the beginning of an entirely new era in computational biology. As AI continues to get better at modeling the complexity of the real world, it will open up new opportunities to cure incurable diseases, treat cancer, and will likely become a core component of any biomedical design cycle. There is also tremendous potential in a field called bioengineering that tries to harness the efficient molecular mechanisms that nature discovered through evolution. Motor proteins, for example, are 4 times as efficient in converting chemical energy into motion compared to a regular gasoline engine. So what if we could engineer an engine based on motor proteins and all it would take to run was a little bit of sugar? Or what if we could use living cells to grow new organic materials with exceptional properties like we see in bamboo or tooth enamel? There's even people working on designing new enzymes that can actually break up environmental waste materials, like hard plastics, that are not traditionally biodegradable. Needless to say that with the ever-growing amount of available data and the sheer complexity of the processes involved, machine learning is positioned as a very powerful tool for many of these scientific disciplines. So in the next few videos, we'll dive into some of the specifics of how to apply deep learning to various areas of computational biology, like protein structure prediction, protein design, property prediction, and much more. So whether you look at it from a purely scientific perspective or you're more of an instinctive type of person, life is undoubtedly the most astounding miracle in the universe, and we are smack in the middle of it. So the next time you're getting some corona blues, just look at your own hand and contemplate, the astounding complexity of molecular machinery that is hard at work to keep you very much alive. Alright, and before ending this video, I wanted to quickly mention that I'm trying out a new platform called Pensight, where I'm available for one-on-one video calls in case you have questions about specific projects that you're working on or any kind of machine learning question in general. Link is in the description. Hope to see you there.